We're talking about the de-extinction, get this, of the dire wolf.
The dire wolf. Wow. Okay.
Yeah.
Yeah, that definitely falls into the category of things you'd think were gone for good. Like, isn't that's like Ice Age stuff, right?
Exactly. You're thinking of those massive wolves, bigger than any wolves alive today, roaming around with mammoths and saber-toothed cats.
Okay, yeah, that's ringing a bell now. So, wait, they brought them back? Like, how is that even possible?
I know, right? It sounds straight out of science fiction, an incredible scientific journey, all the steps they took to bring back this creature.
All right, I'm all ears. So, where do we even begin with something like this? Talk about an ambitious project.
We want to understand exactly how scientists did it,
Yeah.
how they resurrected the dire wolf. All right, so let's jump right in. I guess the first question is, where did this whole crazy endeavor even begin?
Well, like with so much in biology these days, it all started with DNA. Ancient DNA, to be precise.
Ancient DNA. Okay, so we're talking about genetic material from, what, dire wolf fossils?
Exactly. Researchers managed to get their hands on this ancient DNA from dire wolf remains found in two pretty amazing places, actually. One was eastern Beringia.
Beringia. Okay, so that's the land bridge that connected Asia and North America during the last ice age, right?
That's the one. And because of the permafrost, those conditions are perfect for preserving, well, everything, including DNA.
Makes sense. What about the other spot?
The other place was it's probably a site you've heard of, the Brea Tar Pits.
No way! The tar pits. So we're talking about those sticky pits in Los Angeles where they found all kinds of incredible fossils.
You got it. Turns out being trapped in tar for thousands of years is a pretty effective way to preserve DNA too, even if it's not the ideal way to go for the poor creatures that ended up there.
That's a pretty wild thought. Okay, so they've got these ancient bones, and somehow they've managed to extract DNA from them. But I can't imagine that DNA is in very good shape, right? It must be incredibly fragile after all those years.
Oh, you're absolutely right. I mean, we're talking about DNA that's tens of thousands of years old.
Yeah.
It's naturally going to be degraded, broken down into these tiny little fragments. It's kind of like trying to put together a jigsaw puzzle where someone took a hammer to the pieces first.
Ouch. Sounds like a nightmare for the scientists.
Yeah, it's definitely a huge challenge. So to work with this super fragile DNA, the teams had to be incredibly careful. Like they used these ultra clean labs to prevent any contamination from modern DNA, our DNA or bacteria or anything like that.
Right, because even a tiny bit of contamination could mess up the whole thing.
Exactly. And they use these special silica-based methods to gently extract the DNA, kind of like coaxing it out of the bone, without damaging it further. And then get this, they used a process called UDG treatment.
UDG treatment, what's that?
So you know how DNA gets damaged over time? Like certain chemical changes happen. Well, UDG treatment is like a repair crew. It fixes some of those common types of damage. It's like, imagine an old manuscript, all torn and faded. UDG treatment is like carefully repairing some of the tears and making the text readable again.
Okay, so they've managed to retrieve this ancient DNA, and they've even managed to patch it up a bit. But how do they even begin to make sense of it? We're talking about something that's essentially a genetic code, right?
Mhm.
How do they even read that?
This is where things get really high-tech. To read this ancient DNA, they use these cutting-edge sequencing technologies, platforms like PacBio, HiFi, and Oxford Nanopore. These are known for giving you these super long reads of DNA information, which is really important when you're working with those tiny fragments of ancient DNA.
So longer reads, that means they're getting bigger chunks of the genetic code at once.
Exactly. It's like instead of having a puzzle with 1,000 tiny pieces, you get one with, say, 100 bigger pieces. Makes it way easier to see the overall picture.
That makes a lot of sense. But how can they be sure they're actually looking at direwolf DNA and not, I don't know, the DNA of some ancient bacteria that happened to be in the same sample?
Good question. And that's where a technique called metagenomic profiling comes in. Basically, it allows them to identify and separate the DNA sequences of all the different organisms that are present in a sample. Think of it like sifting through a mixed bag of beads.
So they can actually sort out the direwolf DNA from all the other genetic material that might be in there?
Exactly. It's pretty remarkable what they could do these days.
It really is. Okay, so let's recap. We've got the ancient bones, they've extracted the fragile DNA, repaired it a bit, and then read it using these advanced sequencing technologies. So now, what? Do they just have this massive string of genetic letters, and they have to try to figure out what it all means?
That's a great way to put it. And that takes us to the next step, which is called genomic assembly. It's exactly what it sounds like. They're trying to assemble the full genome, put all those DNA fragments back in the right order,
But how do they do that? It sounds like an unbelievably complex puzzle.
It is, for sure. They actually use the genomes of living, related species as kind of a reference. Think of it like using a blueprint to help you put together a piece of furniture.
A blueprint. Okay, that's a good analogy.
The closest living relatives to the dire wolf are the gray wolf,
Mhm.
Canis lupus, and the African wild dog, Lycaon pictus. And because we already have the complete genomes for these species, they can use them as guides to help them assemble the dire wolf genome.
So they're comparing the ancient DNA to the modern wolf DNA and using that to figure out where all the pieces fit. That's pretty clever.
Right. And of course, they use a whole suite of specialized software programs to help with this process. Programs like Fly and Canoe, which are designed to deal with these massive amounts of genetic data. And then, to get things even more organized, to arrange those longer sequences into the right order on the chromosomes, they use a technique called high-C scaffolding.
High C scaffolding. Okay, now we're getting into some real technical jargon.
Yeah, it is a bit of a mouthful. But basically, imagine you have a bunch of notes, and high-C helps you figure out which notes should be grouped together.
So it helps them organize the genome into meaningful sections.
Exactly. It's all about getting that complete picture of the dire wolf's genetic blueprint.
It's amazing how much detail they can get from something as seemingly simple as a string of letters.
Oh, I know, right? But, it's not enough to just have the sequence of those letters.
Right. It's like having a recipe but not knowing what any of the ingredients are or what the instructions mean.
Exactly. So the next stage is what we call functional annotation. And for this, they use all sorts of computational tools. Think of it like having a team of experts who can translate the genetic code.
A team of genetic translators. That's a cool way to think about it.
They use tools like Repeat Masker. The one identifies all the repetitive elements in the genome. Then there's Augustus. It's like a gene finder. It predicts where the protein coding genes are located. And then to figure out what those proteins actually do, they use InterProScan. It's a really powerful tool for understanding the functions of proteins and identifying important regulatory elements.
Regulatory elements. So those are like the on-off switches that control when and where certain genes are active.
Exactly. So at this point, they're not just reading the genetic code, they're actually understanding the language, figuring out what it all means.
That's incredible. Okay, so they've got this detailed genetic blueprint. What can they do with it?
Well, one really cool thing is that they can use it to figure out where the dire wolf sits on the tree of life.
Yeah,
You know?
the evolutionary tree.
Yeah.
So how they're related to other canines.
Right. They use something called comparative genomics. They compare the dire wolf genome with genomes of other members of the dog family, looking for similarities and differences in their DNA.
So how do they do that comparison? Are they just eyeballing it, or is there a more systematic approach?
Oh, it's way more sophisticated than just eyeballing it. They're looking for specific variations in the genetic code. These variations are called single nucleotide polymorphisms, SNPs for short. It's basically a change in a single letter of the DNA code. And by comparing these SNPs between different species, they can trace their evolutionary relationships.
So, like, the more SNPs two species share, the more closely related they are.
That's the basic idea. And they don't just look at SNPs in any old DNA, they specifically look at mitochondrial DNA, the DNA that's passed down only from the mother, and nuclear DNA, which comes from both parents.
So by looking at all these SNPs in both types of DNA, they can get a really good idea of how dire wolves are related to other canines.
Exactly.
So what did they find? Where did dire wolves end up on that evolutionary tree?
Well, what they found was pretty fascinating, actually. The analysis showed that the lineage leading to dire wolves actually split from the lineage leading to gray wolves roughly 5.7 million years ago.
5.7 million years. That's a really long time to be evolving on separate paths. So I guess the next question is, what did all that separate evolution lead to? What made dire wolves so unique? Were they just bigger, scarier versions of gray wolves, or was there more to it than that?
That's a great question, and one that scientists were really keen to answer. So they went even deeper into the dire wolf genome and started looking for clues, basically for genes that might have been involved in the traits that made them so different.
So they were looking for genes that were related to things like their size, their strength, their hunting behavior, maybe even their social structure.
Exactly. And they found some really interesting stuff. First of all, the more they analyzed the genome, the clearer it became that the dire wolf wasn't just some kind of beefed up gray wolf. It was actually quite distinct.
Okay.
Like, based on their analysis of the evolutionary relationships, they proposed that the dire wolf should actually be placed in its own genus.
Wait, its own genus. So not just a different species, but a completely different genus. That's a big deal, right?
Oh yeah, it's a huge deal. I mean, it suggests that the dire wolf was truly unique, not just a close relative of the gray wolf. They even gave it a new genus name, Aenocyon, to distinguish it from Canis, the genus that includes gray wolves and dogs.
So Aenocyon dirus, the dire wolf, it's got a whole new identity now. And did they find any specific genes that could explain some of its unique traits?
They did. They found these groups of genes that showed signs of having undergone positive selection.
Positive selection, meaning that those genes were somehow beneficial to the dire wolf, and so they became more common over time.
Exactly. So these genes give us clues about the kinds of adaptations that made the dire wolf so successful. And one of the things they found were genes related to a hypercarnivorous diet.
Hypercarnivorous, meaning they were serious meat eaters.
Right. Exactly. Like their diet was almost entirely meat. And they found specific genes that supported this. Genes like MSTN, which is involved in muscle growth and maintenance, and AMY2B, which is involved in starch digestion. They also found genes that might have been involved in their adaptation to colder climates, like UCP1 and LEPR, which are involved in regulating body temperature.
So not only were these wolves huge and powerful, but they were also built for a diet of pure meat and for surviving in harsh, cold environments. No wonder they were such formidable predators.
Right. These genetic adaptations really paint a picture of a truly remarkable animal, one that was perfectly suited for its time and place.
Okay, so we've gone from ancient bones to a complete genetic blueprint and a deeper understanding of the dire wolf's evolutionary journey.
We've really uncovered a lot about what made them unique.
But now comes the really mind-blowing part. How do you take all that information and actually bring this extinct animal back?
Okay, so this is where we enter the realm of cutting-edge genetic engineering. Basically, they needed to take the direwolf DNA and use it to create a living animal, but they didn't start from scratch.
Right, because that would be like impossible with current technology.
Exactly. So what they did was they started with a gray wolf fibroblast cell line.
Fibroblast, okay. Those are cells found in connective tissue, right, like skin cells.
You got it. And they chose a cell line that had a really stable genetic makeup, which is important for this kind of work. And the idea was to use these gray wolf cells as a foundation and then basically edit their DNA to make it more like direwolf DNA.
So they're not creating a dire wolf from scratch. They're taking a living relative and modifying its DNA. It's like a genetic makeover.
That's a great way to put it. And to do this editing, they use the ultimate genetic tool, CRISPR-Cas9.
CRISPR. I've heard of that. It's like a super precise pair of molecular scissors that can cut DNA at specific spots, right?
That's exactly it. And they combined CRISPR with a process called homology-directed repair, or HDR. It's kind of like a find and replace function for DNA.
Find and replace. Okay, that's a good analogy.
So using CRISPR, they made cuts in the gray wolf DNA at specific locations. And then they provided these little pieces of direwolf DNA synthesized in the lab that matched the areas around the cuts. And because of HDR, the cell naturally used those direwolf DNA pieces as templates to repair the cuts. Basically, they tricked the cells into inserting direwolf DNA into the gray wolf genome.
Wow. So they're literally cutting and pasting DNA from one species into another.
Pretty much. And they did this for a bunch of different genes and regulatory elements that they thought were important for making a dire wolf, all those genes we talked about earlier, the ones involved in their diet, their size, their cold tolerance, all that stuff. And get this, they managed to replace about 6% of the gray wolf genome with dire wolf DNA.
6%. It doesn't seem like a lot to change an animal so drastically.
I know, it's kind of surprising, right? But remember, they were really targeting those key genes and regulatory elements that they thought made the biggest difference. It's all about quality over quantity, I guess. And they were incredibly careful about making sure that the changes they made were accurate and that they didn't accidentally introduce any unwanted mutations. Like, they did a whole bunch of checks and found that the rate of off-target effects, meaning unintended changes, was less than 0.001%.
That's incredibly precise. It really shows how far gene editing technology has come. Okay, so they've got these cells with edited DNA. How do you go from a few modified cells to a whole animal? That's still a huge leap, right?
It is a huge leap, and this is where it gets really interesting. The next step is called somatic cell nuclear transfer, or SCNT for short. It's the same technique that was used to clone Dolly the sheep back in the day.
Dolly, the world's first cloned mammal. Okay, so this is a pretty well-established technique.
Yeah. Basically what they did was they took egg cells, or oocytes, from domestic dogs and removed their nuclei, which is where the dog's DNA is stored.
So they emptied out the dog egg cell, basically.
Exactly. And then they took the nucleus from one of those genetically engineered gray wolf cells, the one with the dire wolf DNA, and they injected it into the empty dog egg.
So now you've got a dog egg cell with dire wolf DNA inside.
Exactly. And then they activated these reconstructed embryos using a chemical trigger.
So that kickstarts the development process.
Yep. They let those embryos develop in a lab in a petri dish until they reached a stage called the blastocyst stage, which is an early stage of embryonic development.
So it's like a tiny little ball of cells.
Yeah, that's a good way to think of it.
No.
And then they implanted these blastocysts into surrogate mothers, which were female dogs that had been hormonally prepared for pregnancy.
So they put the embryos into dog moms to let them grow and develop normally.
That's right.
And it worked. The dog moms were able to carry these dire wolf embryos to term.
It did. And incredibly, they had a successful gestation period of 61 days.
61 days. That's about the same gestation period as a dog, right?
Exactly. And then the moment everyone had been waiting for, the birth of a dire wolf pup. It was a healthy pup, weighing in at 1.2 kilograms, which is pretty standard for a newborn dire wolf based on fossil evidence.
So after all that work, a healthy dire wolf pup, it's hard to believe it's even real.
I know, right? It's pretty amazing. And then, of course, they had to make sure that this pup really was a dire wolf, so they did a whole bunch of measurements and analyses.
They measured its skull, its teeth, its bones, all that stuff.
Exactly. They compared those measurements to the fossil records for dire wolves. And get this, the match was incredible. They estimated that the pup's morphology was over 99.4% accurate.
Over 99% accurate. That's amazing. So it really is, physically speaking, a dire wolf. What about its behavior? Does it act like a dire wolf?
Well, it's still early days, but the researchers have been observing the pup and learning more about its behavior. And so far, it's been displaying typical canid behaviors, interacting with its caretakers, playing, things like that. And interestingly, it seems to have a strong preference for meat, which aligns with what we know about the dire wolf's hypercarnivorous diet.
It's really incredible. It makes you wonder, what if they could bring back other extinct animals? What if we could see mammoths roaming the earth again, or hear the roar of a saber-toothed cat?
I know. It's definitely a lot to think about.
This whole de-extinction thing, it really pushes the boundaries of what we thought was possible.
It does, and it really highlights how far science has come, in terms of understanding DNA, gene editing, cloning, all of it. It's really a testament to human ingenuity.
Absolutely. And it's also a reminder that extinction isn't always forever. I mean, who knows what's possible in the future?
Who knows? But it does raise some interesting questions, right? Like, is de-extinction something we should be pursuing on a larger scale? And if so, what kind of ethical considerations do we need to take into account? Like, what are the potential risks and benefits of bringing back extinct species? And what about the animals themselves? How do we ensure their well-being in a world that's so different from the one they evolved in?
Those are all really important questions. It's not just about the science, it's about the ethical and ecological implications too.
Exactly. We need to think carefully about the potential consequences of this kind of technology and use it responsibly.
Well said. Well, we've covered a lot of ground today, from ancient DNA to cutting-edge gene editing to the birth of a dire wolf pup. It's been a wild ride.
It really has.
And for our listeners, we hope this deep dive has given you a good understanding of how this incredible feat was accomplished.
Yeah, we've seen how scientists took fragmented ancient DNA and used it to bring back an animal that's been extinct for thousands of years. It's really quite mind-boggling.
And it opens up so many possibilities. So here's something for you all to ponder as you go about your day. If we can bring back the dire wolf, what other extinct species might be next? And what kind of world do we want to create with this incredible power?
The world with dire wolves in it, again, wouldn't be so bad. It's a big responsibility, though, that's for sure.
It definitely is. And we'll have to see what the future holds. Thanks for joining us for another deep dive.
It's always fun.
Until next time, keep exploring.